Okay, very good morning, my dear friends. So, in this regard, CPGET and all JL, DL, Gurukul, DL, JL, and PGT free online coaching. Eminent resource persons are going to deliver their lectures and their efficiency, their subjects, so sharing their knowledge. My dear friends, utilize this wonderful opportunity. So, today's our eminent resource person, Dr.O.Padmaja Madam Garu, Assistant Professor, Department of Botany, Tara, Government Degree and PG College, Autonomous, Sangareddy. Madam is going to explain about the area of the plant sciences, especially embryology, plant embryology. Madam, please welcome and share your screen. I have given to you.
Thank you, sir. Thank you, Tirupati, sir. And students, a very good afternoon. Today we are going to start our session regarding the part of the embryology. Myself, Dr.Padmaja, Assistant Professor of Botany, Tara Degree College, Sangareddy. Today I am going to discuss about the types of embryo sacs, and there are different types of embryo sacs are there. We may expect the questions regarding the types of embryo sac and how the nucleus are arranged in the embryo sac and what is the chromosome number of endosperm in embryo sac. Like that, the different types of questions may be expected from these types of embryo sac. And monosporic embryo sac, they are the types, and what is the difference between monosporic embryo sacs, bisporic embryo sacs, tetrasporic embryo sacs, and our conclusion part. It is a very interesting and the remembering part of this embryology. And first of all, before entering into our topic, we already discussed that embryo sac is also called as female gametophyte. In angiosperms, there are different types of embryo sacs are there, and these embryo sacs are present in the ovule. Ovule is also called as megasporangium. And in megasporangium, the nucellus is present. These nucellus, one of the cell will function as a megaspore mother cell. These megaspore mother cell undergo reduction division. As a result, megaspores are formed. This process is called as the megasporogenesis. Now, how the monosporic embryo sac is formed, bisporic embryo sac is formed, and tetrasporic embryo sac is formed. And before knowing that, first we have to know the difference, why we are calling this as a monosporic embryo, bi, and tetra. The word itself, mono means one, that one megaspore is participating. Bi means the two nucleus of the megaspore mother cells are participating, and in tetrasporic, the four nucleus of the megaspore mother cell. So, first, we already studied that one of the megaspore mother cell undergo reduction division. Reduction division means it is taking place meiosis 1 and meiosis 2. After meiosis 1, there is a wall formation will be taking place, and after meiosis 2, again wall formation. As a result, from one megaspore mother cell, we are getting 4 megaspores. In these 4 megaspores, 3 will be disintegrating. Only one will be function, that is participating in the development of embryos. That is called as monosporic embryo sac. Now we see here. 3 different types of embryo sacs have been reported on the basis of number of megaspore involved in the development of embryo sac. They are mono, bi, tetrasporic. Now, in monosporic embryo sac develops from a single megaspore. Only one megaspore is participating in the development of embryo sac. In that, development, there are two types are there. One is polygonum type, another is Oinothera type. And polygonum is considered as a primitive type of embryo sac. And in polygonum, this type of embryo sacs develop from the chelazal megaspore. Just we have to remember that chelazal megaspore will be functional and its nucleus divides thrice. Thrice means 3 times to form 8 nucleus. That means once you see here, female gametophyte is haploid, the nucleus is haploid. The haploid nucleus undergo 3 times mitotic division. As a result, 8 nucleus are formed. So this type is generally referred to as normal type of embryo sac. And it is also called as polygonum. In the polygonum plant, first it was discovered. So it is called as a polygonum type of embryo sac, and it was studied by the Strasburger. And now, if you see the diagram, here, first one is the megaspore mother cell. This megaspore mother cell undergo reduction division. Reduction division, we are having the meiosis 1 and the meiosis 2. After meiosis 1, 2 nucleus are formed that are called as dyad, that upper dyad and the lower dyad. And again, this one will divide by meiosis 2. As a result, 4 nucleus are formed. In these 4 nucleus, if the chelazal one is functional, if you see the diagram, chelazal side is a functional megaspore, and the upper 3 are disintegrating. That means the megaspore that are present in the chelazal is functional, and the remaining 3 are disintegrating. The functional megaspore will contain the haploid nucleus. That means by the diagram itself, it is showing that megaspore mother cell undergo meiosis. As a result, 4 megaspores are formed. By the wall formation, we are clearly seeing there. And the 3 that are present in the micropylar end, they are disintegrated. That means they loses its identity, and only one that is present towards the chelazal end will enlarge in size and contains the haploid nucleus. This haploid nucleus now undergo, that is called as thrice. One mitotic division gives rise to 2 nucleus, and 2 mitotic division give rise to the 4 nucleus, and 3rd mitotic division give rise to the 8 nucleus. And okay, is it audible?
Yes,
ma'am.
Okay, thank you. Now, here we are seeing that one of the nuclei will be moved to the center, and another from the micropylar will move to the center. As a result, a secondary polar nucleus is formed here. So this is called as the polygonal type of embryo sac. It is come under the monosporic type, and that is also the functional megaspore is at the chalazal end. The 3 points we have to remember here. And after fertilization, the endosperm is 3N in condition. And next one is the Oinothera type. In Oinothera type, here it is a monosporic type of embryo sac. And if you see the diagram here, after the megaspore, mother cell undergo reduction division. As a result, 4 are formed. And in these 4, 3 chalazal end will be disintegrating. Only the functional megaspore at the micropylar end will be remaining. And this will be enlarged in size, and it contains a haploid nucleus. So the female gametophyte is haploid. And this haploid nucleus undergo 2 mitotic divisions. Once you see, after 1 mitotic division, 2 nucleus are formed. Again, the 2 nucleus undergoing another mitotic division. As a result, 4 are formed. In these 4, the 3 will be functions as agaperators, and only 1 will be remaining at the center. And this is the polar nucleus. So after fertilization, what happens? The antipodals are absent here, and endosperm will be diploid in condition. So the chromosome number in Oinothera is diploid, is the endosperm. And now here the 2 examples come under the monosporic type of embryo sac, that is Polygonum and Oinothera. And the difference is, in Oinothera, the micropylar is functional, and in Polygonum, the chalazal is functional. And the endosperm is diploid in condition in Oinothera, and the endosperm in Polygonum is diploid in condition. That we have to remember for the bits. And the next one is the bisporic embryo sac. In this bisporic embryo sac, this megaspore mother cell undergo meiosis 1. As a result, the wall formation will be takes place, and the upper are called as the dyads. So the bisporic embryo sacs develops from one of the 2 dyads formed as a result of first mitotic division of megaspore mother cell. One of the dyad, in that the one of the dyad will be disintegrate, and only one of the dyad will be functional, and that from functional one, if the embryo sac is formed, that is called as a bisporic embryo sac. That means the dyad is taking part in the formation of embryo sac. Now, both of the nuclei of the functional dyad, just you have to remember that in dyad 2 nucleus will be there, but there won't be any fold formation. So that is called as the dyad. And this dyad will having the 2 nucleus that is participating in the embryo sac. That condition is called as bisporic. Bi means 2 nucleus are present in the dyad, and the formation of the embryo sac will be taking place. And each nucleus undergo 2 mitotic division, and as such, the mature embryo sac is 8 nucleate. And in these 8 nucleate, the 8 nucleate are organized into antipodals, agaperators, and polar nucleus, just like a polygonal type of embryo sac. On the basis of position of functional dyad, the following 2 types have been recognized in the bisporic embryo sac. That means the which one is functional. On that basis, they are of 2 types. One is allium type, another is endymion type. In allium type, develops from the chalazal dyad. That means chalazal dyad is functional, and that dyad will be participating in the embryo sac development. And in endomion, micropylar will be functional, and that from the micropylar dyad, the embryo sac is developed. That is called endomion type. These 2 come under the bisporic embryo sac. Just by the diagram, if you see here that the megaspore mother cell, this is the dyad formation. In this dyad formation, if the upper dyad will be, the micropylar will be the disintegrating, and the chalazal will be functional, and this dyad will undergoing the 3 mitotic division. As a result, 8 nucleus are formed here, and these 8 nucleus, just they are present as the same as that of the polygonum type. Here the micropylar and this is the egg apparatus is present, and the chelazal and antipodals are present, and at the center two polar nucleus or the secondary nucleus are present. And after fertilization, again here the endosperm is rearing condition and the zygote is diploid, and this diploid zygote undergoes into embryo, and these synergies will be disintegrating. And next one is the tetrasporic type of embryo sac. In tetrasporic type of embryo sac, we can also call as here the cenomegaspore. Ceno means if more nucleus are present, that condition is called as cenocytic or ceno. Here the megaspore consisting of 4 nucleus are present. So that is called as a cenomegaspore. That means here megaspore mother cell after meiotic division, meiosis 1 and meiosis 2, there is no wall formation. 4 nucleus are present in that megaspore. So it is called as cenomegaspore. If the cenomegaspore participate in the formation of embryo sac, this type of embryo sac is tetrasporic embryo sac. This tetrasporic embryo sac, there are following types are there. They are recognized in the tetrasporic. Just we have to remember how the mitotic divisions are taking place, and what are the nucleus and are present in the matured embryo sac in the tetrasporic type. So the first one is the Edoxa type. In this Edoxa type, just you see the development of the megaspore mother cell. Here it has 8 nucleus which are formed by the mitotic division of the nucleus of the cenomegaspores. That means here the 4 nucleus, in this 4 nucleus undergoing the mitotic division, as a result 8 nucleus are formed. Here the 4 nucleus are participating without the wall formation. So that condition is called as the cenomegaspores. And here the first one is the Edoxa type. The arrangement of 8 nucleus in embryo sac is same as that of the Polygonum type. So this type of embryo sac occurs in the Edoxa, Sambucus, Almas, and Tulipa, and Erythronium, etc. And in this also, after the fertilization, the zygote will be formed and the endosperm will be formed. This endosperm is the triploid condition. So that we have to mainly concentrate on that. What is the chromosome number of the endosperm and what are the nuclei that are present in the mature embryo sac. And next one is the plumbago type. In this type, of the 4 haploid nucleus of the cenomegaspore, one migrates into the micropylar end, and one of the chelazal end, and 2 onto the 2 lateral sides. Each of these 4 nucleates divides again, which results 4 groups of 2 nucleus each are formed. And if you see the diagram, we can clearly understand what are the nucleus that are present in the micropylar end and the chelazal end. One of the nucleus from each group moves to the center of the cell, 4 polar nuclei. The remaining nucleus at the micropylar end cut by the membrane and formation of the egg. If we see here, and next one is the pinia type of embryo sac development. In this pinia type of embryo sac development, if you see the matured embryo sac, by this matured embryo sac, we can clearly understand here how many nucleus are present. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. That means in matured embryo sac, 16 are present. And how these 16 are formed from the cenomegaspore. That means we already know that here the megaspore mother cell undergo meiosis 1, as a result 2 are formed, and another meiosis 2, 4 are formed. This condition is called the cenomegaspore, and this cenomegaspore is itself is having the 4 nucleus. In order to get the 16 nuclei, it have to undergo to mitotic division. The 4 after mitotic division give rise to 8, and these 8 after mitotic division give rise to 16, and these 16 are arranged like this. That in the micropylar end, egg apparatus is present. Egg apparatus consisting of a center egg nucleus, and on either side synergids are present. And at the lateral side, lateral means on either side of this embryo sac, 3 nucleus are there on either side of this embryo sac on the lateral side, and at the antipodals, the 3 are present at the chalazar and the 4 nucleus are migrated or moved to the center. And these 4 haploid nucleus, after fertilization, another male gamete will fuses with this by the fusion. As a result, the endosperm is formed. Here the endosperm is 5N in condition. That means pentaploidy condition is seen in the endosperm on the pinia. So in this type, 4 haploid nucleus of the cenomegaspore undergo 2 successive mitotic division, forming 16 haploid nucleus. In this 16 haploid nucleus, 3 nucleus arrange themselves in 4 groups of 4 each. One at the micropylar end, one at the chalazar and one at the 2 lateral side. That means the second diagram, if you see on the micropylar, there 4 are present, and in the chalazar 4 are present. On the lateral side also 4 are present. And what happens in this? One is migrating from the micropylar end towards the center, and one is migrating on either side of the lateral side to the center. As a result, 2 are forming, and at the chalazal end again one will be migrating at the center. As a result, 4 nucleus are there here, and these 4 nucleus, as a result of these, but a male gamete fuses with these 4 nucleus. As a result, a 5 nucleate endosperm is formed in this embryo sac. Now, 1 nucleus from each group migrated to the center of these 4 nucleus in the form of 4 polar nucleus. That means in this, the polar nucleus 4 are present in the pinia type. The remaining 3 groups of nucleus, 1 chalazar and the 2 lateral, disintegrate, and afterwards these will be, the lateral side will be disintegrating and the antipodals will be disintegrating, synergids will be disintegrating. Now the embryo sac contain only the fertile structure of after syngamy is zygote is formed, and after the double fertilization the endosperm is formed. That is the pentaploidy condition structure embryo sac is seen here. And now here the another type is the peperomia type. In this peperomia type, if you see the diagram itself, this is called as the third diagram is called as the cenomegaspore. And how the cenomegaspore is formed? The megaspore mother cell undergo meiosis 1 without wall formation, meiosis 2 without wall formation. As a result, the megaspore contains the 4 nuclei, and this 4 nucleus structure again undergoes the mitotic division. As a result, if you see the matured embryo sac, by this matured embryo sac, how many nucleus are there? Like this, that how many mitotic division have to take place, we have to see here. That means once we look at the documentation of this, the 4 haploid nucleus of cenomegaspore undergo 2 successive mitotic division, forming the 16 nucleus. That means cenomegaspore 4 are present. In order to get 16, it have to undergo 2 mitotic division. How? The 4 will undergo 1 mitotic division. As a result, 4 plus 4, 8 will be formed. Again, that 8 mitotic division is will taking place. Double the number, that is 60 will be forming here. So, of these 2 nuclei at the micropylar end form the egg and the synergids. That means you see here in the micropylar region, only the egg and the synergids are present. Only single synergid is present and the egg cell is present. And here 8 fuse in the center of the cell to form the polar nucleus. That means at the center 8 nucleus are present and 8 fuse to form a polar nucleus. The remaining 6 which are cut off by the membrane at the chalazal end may be compared with the antipodals. That means that are present at the chalazal end, they may be formed as antipodals. And after fertilization, and what is the fate of this embryo sac is, One male gamete will fuses with the egg, as a result zygote is formed. Another will fuses with the polar nucleus, which contains 8 nucleus are present. Another male gamete will fuses, as a result 9 end structure endosperm is formed here. After fertilization, the synergid will be disappeared and antipodas will be disappeared. Only the zygote and the endosperm is formed. And what is the chromosome condition of endosperm is already 8 are present. Another male gamete will fuses with this. As a result, 9 end condition of endosperm is formed here in peperomia type. Now, here we see the drusa type. So, like pinia and peperomia type, this is also a 16 nucleate embryo sac. If you see the embryo sac, if you count the nucleus, they are 16 are present. And that means the arrangement somewhat will be the changes, but the embryo sac is 16 nucleated. And if you see how the stages are seen here with the tetrasporic embryo sac, that means megastore is present, and this megastore, the 4 nucleus are there. These 4 nucleus undergo 2 mitotic division. By first mitotic division, 8 nucleus are formed, and by the second mitotic division, 16 nucleus are formed. And if you see that in matured embryo sac, 3 nuclei form the egg apparatus. That means in the micropylar end, egg apparatus is present. Egg apparatus consisting of at the middle egg and on either side, 2 synergids are present. And to act as the polar nucleus and the remaining 8 nucleus, only 2 are present at the center. They are acting as a polar nucleus. And remaining 8 nucleus are forming at the antipodals. 11 plus 2, 13, 14, and the egg apparatus, 14, 15, 16. Like this, total embryo sac consisting of 16. In this 16, the antipodals contains the 11, and the polar nucleus contains the 2, and egg apparatus consisting of the 2 synergids and the egg. So this type of embryo sac is present in Drusa, Rubia, Chrysanthemum, and Almus. And here, if you see here, the antipodals are 11 in number, and after fertilization, these antipodals will be disintegrating, and 2 synergids will be disintegrating, and the egg and the male gamete, one of the male gamete fuses with the egg, as a result, zygote is formed, and the polar nucleus consisting of 2 nucleus. And after double fertilization, the one of the male gamete fuses with the polar nucleus. As a result, here the 3N condition endosperm is present in the drusa type. And next one is the fritillaria type. Here somewhat will be taking place like that. Here the 4 haploid nucleus of the megastore arrange themselves in the 2 groups. Here 3 at the chelazal end in the form of a triploid nucleus and 1 at the micropylar end. If you see the third diagram here, that one will be remaining at the chelazal end, 1 will be remaining at the micropylar and 3 will be remaining at the chelazal end. And the triploid chelazal as well as the haploid micropylar nucleus undergo 2 mitotic division. As a result, 4 triploid nuclei are formed at the chelazal and 4 haploid at the micropylar end. That means you see here on the upper side, 2 are formed, and the lower side here that 1 give rise to, in each one, 1, 2, 3 nucleus are present here. That is called the triploid one. And the remaining 1 haploid and 1 triploid nucleus move at the center. Just we have to remember that here the triploid means 2, 3 nucleus are present here. And 1 will be haploid and 3 will be triploid structure will move at the center. That means how many are there? 3 plus 1, 4 are present at the center. And the egg apparatus consisting of egg and the synergids. And at the antipodal, 3 triploids are present here. So remaining one haploid and one triploid nucleus move to the center when they fuse to form the tetraploid polar nucleus. Just here, what we have to concentrate here in the antipodals, that each one is a triploid one. Each one is a triploid one. Here one triploid, here three triploids are present here. And at the center, one haploid and the triploid nucleus is present. Triploid nucleus consisting of three nucleus are present in a triploid cell. Triploid means three nucleus are present and one haploid. The total polar nucleus consisting of 4n in condition. So after fertilization, here one of the male gamete will fuses with the egg. As a result, zygote is formed. Another male gamete will fuses with the polar nucleus which contains one haploid and one triploid nucleus. Triploid means two nucleus are present. So that triploid nucleus, as a result, total 5n condition of endosperm is present, and after fertilization these synergids will be disappeared and this egg will be developing into the embryo, and at the center it will be developing into the endosperm, and these triploid antipodals will be disappearing. So here in fritillaria type, here the triploid type of structure is formed by this division. So at last, the embryo sac consisting of 16 nucleus. In the 16 nucleus, 3 are present at the micropylar end and 4 are present at the center and 9 are present at the antipodal. In this 9, each one is considered as a triploid. One triploid, two triploid, three triploid. Each triploid consisting of 3 nucleus. 3 into 3. It means total 9 are present here. So here the triploid structure of cells are present here in the fritillaria. Next, Plumbazella type. In this Plumbazella type, just like the fritillaria only, but here the initial development of this type of embryo sac similar to the fritillaria type. A triploid nucleus is formed at the chelazal end and the haploid at the micropylar end, just like the fritillaria. And each of these nucleus undergoes single mitotic division. Only single mitotic division will be taking place as a result and forms two groups of 2 nuclei each. One haploid nucleus from micropylar end, one triploid nucleus from the chelazal end, fuse at the center of the sac to form a tetraploid polar nucleus. Tetraploid means 4. And one haploid nucleus at the micropylar end forms the egg, and one triploid nucleus at the chelazal end single antipodal. There are no synergids. Here in this, on either side of the egg, no synergids are present. And only the haploid nucleus is present. And at the center, there is a haploid nucleus and the triploid one. And after, and the antipodals and one triploid will present at the antipodal, at the chelazal end as antipodals. And after fertilization, one of the male gamete will fuses with the egg. As a result, zygote is formed. Another male gamete will fuses with the polar nucleus. The polar nucleus consisting of 4 nucleus. So by adding another male gamete after fertilization, now the endosperm condition is 5n. So it is pentaploidy condition. So they may be asking the question, what is the chromosome numbers in this type, in this Plumbazello or fritillaria or drusa type? Like that the questions may be asked in this regarding this the embryo sac. And this is the overall diagram representation of different types of embryo sac, what we have learned just now. So this is the glance. By this glance, you can understand which type of embryo sac will come under monosporic, which type of embryo sacs come under the bisporic and which type of embryo sacs come under the tetrasporic, and what is the chromosome number in the endosperm, in the angiosperms. So the remaining type of questions what we have learned just now, total glance will be present in this diagram. So if you see that they mainly concentrate on which one mainly concentrate on the endosperm, the chromosome number of the endosperm, and if the xenomegaspore is present, and how many the mitotic divisions are taking place, and in matured embryo sac, how many nucleus are present. That type they may be the questions will be asking in the entrance. If you have doubts, you can ask me.
Madam, good afternoon.
Good afternoon, sir. Good afternoon.
Madam, your explanation is very clear cut, madam. But my request is when you are sharing this PPT in the WhatsApp group, it is very good to remember the points. Actually, here different type of monosporic, several types are there, bisporic, several types are there, in tetrasporic, almost nearly 6 to 7 types are there. So it is very difficult to remember. That's why share this PPT in the WhatsApp group. Remember,
No, it's a voice are breaking, Vijay. One second, you voice.
Madam, my request is please share the PPT in the WhatsApp group. Madam.
Okay.
It is become to number of times we are repeating. Okay.
Sure. Okay, compulsory. I will send the. Sure. And at the same time, tomorrow
This is my humble request, madam.
Sure, Vijay. And what you have to do, the students, is after completing the embryology, there may be one test will be there in the regarding the embryology. So who are going to score good marks, there may be some incentives will be there for the students. This is on behalf of ourselves. So what we are telling, no, you have to concentrate on that. And after completing total embryology, one test will be there. That test questions will be from previous questions. The previous questions of different question papers that are come under during the JL or DL or any entrance. That type bits we will be gathered at that time. So compulsory, I will share the PPT. Hello?
Madam.
Yeah, daily all the students are participating in this coaching.
Yes, madam.
Okay. After completing these classes, are you spending some time regarding to recollect the what are the classes we completed? Are you spending time for recollecting the classes again?
Yes, madam, definitely. Especially your classes. It's very clear cut. The PPT is very useful to us, madam. It is very clear cut and minimize each and every corner. When we see the PPT, we can easily analyze. We can easily remember the things.
Yeah. That is what you are telling is correct. That means today what we will learn after 5 days or 6 days, the memory power will be somewhat we can't remember that. We can't remember. That is the nature of the human. So if you once you by one glance, you can remember.
Yes, that's why I'm asking the notes. Without notes, we can't do anything.
Sure. Yeah. Correct. What you are saying is correct.
More and more.
Sure. PPT. That means everyone is. Once again, you repeat now question.
Madam, as soon as possible, you will share the notes.
Okay. Sure, Prakash, Vijay,
Thank you.
Manoj Kumar. Okay, Anna. Okay, thank you.
Madam,
Acha.
madam,
Tell me, Anna.
at the beginning, this embryology from first class, I think it did not share the PPD in group.
Yeah, yes. What you are saying is correct.
I
Correct,
didn't,
Prakash.
some of the
From the
points I heard, but I didn't listen the class because of the network issue. So
sure,
I
sure.
seen some risk on that day, so I did not listen.
Okay. Total 4 classes PPT I will send you. Okay.
Okay, ma'am, thank you. And
Okay.
according to this and today's class and previous last week class, you share this PPT in group.
Previous class?
Previous class and today's class. Now told now this PPT share in group, madam.
Okay. Sure. Compulsory.
Okay,
Madam,
ma'am.
And
one more request, madam. Madam, here from the microsporogenesis and male gametophyte, female gametophyte, development of the microsporogenesis, megasporogenesis, and where the questions are raising. First of all, in the
main questions, then the main questions, what are
Yeah,
the
madam.
questions they may be asked is just that they won't concentrate on the megasporogenesis or microsporogenesis. They will concentrate on the new cellers. What is the chromosome condition of the new cellers,
Yes, madam.
and in a monosporic embryo sac, they will do the just like a match the following, or they won't do direct questions. That means that may be related to 3 or 4 questions. They will combine and give as a one question. That means you have to concentrate, that you have to logical thinking must be there. That means in which type pentaploid type of endosperms is present, and which one will come under the bisporic type of embryo sac. In bisporic embryo sac, what is the chromosome number of the endosperm? That mainly endosperm they will be concentrating on that. And in the microsporogenesis, they mainly asking the questions like how many celled stage these pollen grains are released,
Shaded.
and they also concentrate on the syngamy, and what is meant triple fusion, and who discovered that one, and related to here, ovules, in which family this type of ovules are seen. For example, in Asteraceae, you already know it is a well developed family in dicots. In this Asteraceae, which type of ovule you can see, and in gymnosperms, which type of ovule? That means the gymnosperms is somewhat primitive when compared to angiosperms. So in gymnosperms, there is a primitive ovule is known as the orthotropous ovule. That is a straight ovule. In this type related questions, and in angiosperms, in which family you can see? That means one time they ask the question that in Fabaceae, which type of ovule is seen? That is campylotropous ovule. And in xerophytic condition, which type of ovules will be seen? They will give the example. They will give the examples like a cactus and some opuntia, like that. And another bit is all. So if you know that all 3 will belongs to xerophytic, you can easily put that question, that is the carcinotopous type of ovule is present in the xerophytic. And in a legumes, like they will use the Dolichos lab and Arachis hypogaea, and next one is any Fabaceae member, like that, in which you can see this acampylotropous means. If you know all this, or otherwise they will do the Fabaceae or another family, like that type of related to the taxonomy and related to the embryology, that like that they will do the questions. That means you already concentrate on the taxonomy and the embryology. If you combine with this, like that, analogical thinking will be useful. So
Thank you,
mainly,
madam.
okay, Anna, okay.
And one more thing, madam, after completion of the session, entire session, you just focused on the especially headings and examples, madam. I think this is very much important For
Okay,
example,
okay.
monothecus, dithecus, monothecus uses
Yeah,
for angiography.
Yeah,
thecus,
monothecus, bithecus, dithecus, and the monothecus, which type of family you can see. And is also another question is, if only one pollen tube is formed, monocypherous condition, and if many pollen tubes are formed, this is polycypherous condition. That way will be considered. So what I am thinking is, after completing whatever the bits that will be coming to the embryology, that we will discuss one day. So after that we will close it.
Yes, madam. One session is conducted for the simplification.
Only
just
simplification and only
simplification.
what we
Examples.
have to concentrate total embryology.
Yes, madam.
That will be best. So before going to get that knowledge, you have to concentrate on this theory part. If you will
Yes.
concentrate on theory part, means you will get that interest. So where I have to learn and how I have to put that answer correctly, like that you will get that idea.
Yes, madam. Definitely, madam. We will work on it, madam.
Okay. Okay. And you also know that the last year మా స్టూడెంట్ అనమాట, తను కూడా ఎట్లా అంటే తన క్లాస్ అయిపోవడం మేము వెంటనే బిట్స్ చెప్పేవాళ్ళం. బిట్స్ అయితే చాలా కాన్సన్ట్రేట్ చేసేది. షీ గాట్ ద ఫస్ట్ ర్యాంక్. కంపల్సరీగా మా స్టూడెంట్స్ అనమాట, అంటే అలా నేను చెప్పొద్దు కానీ మా కాలేజ్ లో ఉన్న స్టూడెంట్స్ ప్రతిసారి క్యాంపస్ సీట్స్, ఉస్మానియా క్యాంపస్ లో 2 సీట్స్ వస్తాయి. ఫస్ట్ ర్యాంక్, షీఈ్ ద ఫస్ట్ ర్యాంక్. ఇఫ్ యూ నో దట్, మీకు ఎవరైనా తెలిసిన వాళ్ళు, మీ సీనియర్స్ ఎవరైనా ఉండేది ఉంటే, షమీమ్ అనే ఫస్ట్ ర్యాంక్, రోహిణి అమ్మాయి 7th ర్యాంక్. వీళ్ళు లాస్ట్ ఇయర్ బిఫోర్ లాస్ట్ ఇయర్ వాళ్ళకి ర్యాంక్స్ వచ్చినాయి. ఏంటంటే వీళ్ళు ఇలానే కాన్సన్ట్రేట్ చేసేవాళ్ళు. పిల్లలకి ఇంట్రెస్ట్, క్లాస్ అయిపోయిన వెంటనే బిట్స్ అడగండి మేడం, బిట్స్ అడగండి అనడం వల్ల ఏంటిది అంటే, వాళ్ళ యొక్క ఇంట్రెస్ట్ ఏమవుతుంది అంటే, మాకు కూడా చాలా రేపు పిల్లలు ఇంట్రెస్ట్ చూపిస్తున్నారు కదా అనేసి అంటే, ఎవరైతే ఖచ్చితంగా కాన్సన్ట్రేట్ చేస్తారో యు ఆర్ గోయింగ్ టు గెట్ ద ర్యాంక్ అండి. ఎందుకంటే మనకు కావాల్సింది ఏంటంటే క్యాంపస్ సీట్స్ కావాలి. క్యాంపస్ సీట్ కొట్టినప్పుడు మన లైఫ్ టర్న్ అవుతుంది. ఆర్ అదర్వైజ్ యూ కెన్ గెట్ ద సెంట్రల్ యూనివర్సిటీస్ లో కూడా మనం పెట్టవచ్చు ఈజీగా.
Yes, madam. We are also comes from Telugu medium background. So
No
మీరు
problem.
ఒకసారి తెలుగులో కూడా ఎక్స్ప్లెయిన్ చేస్తే మాకు అది కొంచెం ఎక్కువగా పెనిట్రేట్ అవ్వడానికి కూడా ఛాన్సెస్ ఎక్కువగా ఉంటాయి.
ఓకే ఓకే ఓకే.
సేమ్ మీరు కూడా సేమ్ వాళ్ళకి ఎలా అయితే క్వశ్చన్స్ ఫ్రేమ్ చేస్తారో మాకు కూడా అలా క్వశ్చన్స్ ఫ్రేమ్ చేయండి.
ఖచ్చితంగా విజయ్, ఖచ్చితంగా.
థ్యాంక్ యూ మేడం.
ఓకే అన్న. ఓకే.
Right. Thank you very much, madam. Okay. 1:30, our next session, sharp 1:30. We will meet again at 1:30 p.m., my dear friends. Okay?
Okay, sir. Thank you.
Okay, sir. Thank you, sir.
Right.